Thank you, everyone, for joining us here. I'm Coach Stanistic. We will do a very brief introduction of the members here. I myself, Roger Nilsson, Lucia, and my graduate student, Erica Kunk, will be with us today. After the brief introduction of who we are, in case some of you do not know us, we will be telling a little bit about what the goal of the workshop is, and then give you a little bit of a perspective of historical perspective of how these things develop at the EarthChem and everything, and eventually we'll tell you that how you can not only upload your data to this site, but also you can use this data for your research if you're an experimenter, as in my case. That's what I do. So I'm Gotri Smisik. I did my PhD in 2009 at University of Cincinnati, mostly thermodynamic modeling. And then after that, I did two postdocs, one at Stony Brook with Don Linsley and Hannah Necklasil, where I switched to be an experimental petrologist, a little bit more from numerical modeling. And then I was at Lamont and American Museum of Natural History till 2016, until I accepted a position here at South Dakota Mines as a petrologist. I'm an igneous petrologist by training, experimental petrologist. Mostly my research involves the processes that happens in the upper mantle, lower crust, and also this planet and different planets such as Moon, Mars, and other terrestrial bodies. So having said that, I should pass to Roger.
So Lucy, why don't you fill people in? You're the chief operating officer of this operation. Why don't you talk about what your role is and how you can help people?
Okay. Hi, everyone. My name is Lucy Perfetta. I'm the product manager for EarthChem and one of the data curators for the EarthChem library repository. And through EarthChem, we've set up a community to host experimental petrology data sets and to make it easier for people to reach both TraceDs and leopard data. And that's pretty much what Roger and Gotcha are going to be introducing you to today. So you can hand that back to Roger.
I saw Paula just popped in. Do you want to give a real quick background on what
Roger doesn't want to introduce himself. That's the
No, I'm saving the best for last. So can you hear me, Paula?
I didn't catch what background you wanted.
Just what's your role in the databases, et cetera, the work that you've done with me, for example.
Oh, I see.
EarthCube.
Yeah, I've been updating the leopard TraceDs database and upload site to updating it for the new schema and template for the TraceD specifically, and adding in a lot of the data for mixed volatiles from the Gios and Gualda compilation, calibration, and also then working to transfer this from OFM, where it was previously hosted, to EarthChem, and then
which we will tell to the group about all that.
Yeah, exactly. But yeah,
so I'll pull all of this together.
Erica,
Erica is the next for speaking of uploading. So why don't you briefly just say what you've been working on, Erica?
So I've been working on sorting the data, the TraceD data, and basically putting them into compositional graphs so we could look at different dependencies.
Right, but for these
or-
people, the critical thing, Eric, is what you were doing in terms of transcription and
Oh yeah.
facilitating upload. Yes.
Okay. So I've been looking at new papers or present day papers and just basically taking the data from these papers and putting them into these Excel sheets, which can be updated onto the tracees.
Right. So where that ties in is that Paul is developing a new interface for upload. But in a historical perspective, this started over 20 years ago when a group of people related to the Germ initiative wanted to have a tabulation of the reservoir components as well as partition coefficients. So they talked me into putting together the Germ partition coefficient database, which most of you have probably used. But in the last 10 years, the emphasis has switched to having a repository for the experimental data that exists so that it's more accessible to the entire community. The problem with the old Germ database was it was basically a summary of the existing data that was compiled by me with my own bias. And what we realized at the end of that, in about 10 years ago, that what we were doing was essentially facilitating people's bad behavior at how they were choosing the partition coefficients they were using in modeling. So we began, Mark Yorso and I began working on tracees. Of course, Mark had worked at the consortium to develop LEPR. And so Mark and I began that work to actually produce a repository. What's happening now, as Paula mentioned, is a lot of this data is now going to migrate to a new interface at Lamont under EarthChem. So that's one of the reasons why we're doing this tutorial and workshop today, is to begin to acquaint the community with what's actually happening. So towards that end, I'm going to go ahead and pull up on my screen that site. So this is the site that Lucy and her group have put together, the community website. So can everybody see that?
Yep.
So this is the experimental petrology community site. And you can see that there are tabs here that will eventually all plug you into how to, for example, search. Eventually this interface will be linked to the search engine for tracees and LEPR. However, the first thing we want to talk about and begin a community discussion on are the submission guidelines for what data are to be included. It's basically the minimum requirements in the metadata. So Gutches, can you go over this part of it, and then I'll do the aspirational.
Yes, you sort of like changed the style of this. We want to remind people that the LEPR and the tracee was under the OFM. Now, as you guys see that it is under the EarthChem, under this website. So that's one of the things. And the why we are here today was able to show you how to upload your data, how to use your template format. And when you are done with it, who to send and who to contact. But more importantly, how can you extract the data to use it within a context for your own research? So here are our minimum requirements. So what do we upload here? We upload only experimental data, not physical measured natural data. Two, we upload only the data from the published material. So it needs to be peer reviewed. And the minimum requirements for that is we need your run temperatures, pressure, time, if you are doing time series experiments, what sort of a device you use, piston cylinder, capsule material, so that we can define the FO2. And then not only the starting composition, but the major element composition of the each phases, as well as the trace element compositions, not only the partition coefficients, but the abundances, so that people can themselves, if they feel like that, they can calculate the partition compositions. Obviously, when you are reporting your trace element contents, it would be great if you can give us the mineral liquid pairs, so that that's one of the things that we are watching out because of the fact that how the trace elements in an experiment act different than a regular phase equilibrium system. And we are hoping that we will have some error measurements for each of these things. And it's in a way, we are requiring those minimum things so that in our metadata, so that when which we will show you in a minute, that we have for all of the minerals that are interested in, we produce this, and then we put together all of the experiments from lots of people, so that you will be able to take them and use it for your own research without digging into the papers to figuring out where those informations are.
So as Erica can tell you, one of the challenges that we have in doing this is that experimentalists don't all report things in the same way or report the same things, period. So it provides a lot of challenges for anybody who's transcribing the data to be able to get in a coherent form to upload it. So part of the conversation we want to begin to have is, how do we agree to a greater extent on what actually gets published in the papers. We also have a set of sort of aspirational goals, and this is coming from what NSF has been asking from the experimental community. And those are listed under the recommended metadata. Specifically things such as images associated with the analyses, so that not only do you know what the number of analyses are, you have specific information on the spatial association. We can't get there yet, but it's a sort of thing that most experimentalists are producing, and some of them are actually publishing them, but they're not being published in a way that's easily transcribable into a database.
And in a way, with that aspirational goal, our hope is when we all publish the research we do, we put all of the important things about that story into the tables and the figures. But the rest of all of the stuff, the VSC images, everything is always in an Excel document with the data points. And when the journal wants, okay, the supplementary material, we dump the rest of it to there. The goal is hopefully with this format that we are producing, you will be able to put it into there rather than just dumping Excel to the journal, so that journals will be accepting without giving too much trouble with how the supplement needs to be. And you can refer that this is the NSF accepted EarthCare format, so nobody can argue with you on that.
So at this point, are there any questions that have come up with just the submission guidelines and the minimum versus aspirational data?
I don't know if this is a question or a comment. Is it okay just to speak out?
Yeah.
Absolutely.
I look at the recommended metadata and I see analytical error, and you're telling me that's due to counting statistics, which I'm totally happy with and understand what that means. And I can put some level of significance on that if I like to. But if I look at error for each measurement, I can interpret that a variety of different ways. What do we need? And do we need to define this better? Is this negotiable? Or do I just need to tell you, this is how I interpret my error and this is the value?
The important thing is to make sure everybody knows what the error is that you have just put in your table. So that's the most important part. The consensus, as you know, is that people publish the 1 sigma standard deviation of averages. That's that's the normal way people are doing that. There are description notes in the template that will allow you to put information in on that. But this is, again, one of the reasons why we're having this conversation, because the largest source of problems with the trace element database is contaminated analyses. Basically, where you thought you were hitting a mineral, but you were actually hitting a mineral plus glass. That's not something that was going to show up. Well, what will happen when you average the data is that problem averages out. And if you're near the detection limits, it's even worse in that the only data that you're going to accept, the non-zero values, are probably contaminated. So we need to do a much better job at doing exactly what you said, which is being clear about how we're calculating those errors.
So to answer your question, Phil, is yes, you can report. I report, for example, county statistics. All you need to tell is there's a place that you can explain what you're reporting so that people will know what it is.
So it's a problem I think most of us recognize. And even now, the template doesn't completely solve it. It basically is designed to report on what you publish. So anything that's gone through the peer review, hopefully they have reported on what type of error is that they're actually reporting. But I don't know that, does that actually answer your question, Phil?
Sure, it absolutely does. Yep.
So are there any other questions about the minimum required metadata versus aspirational?
I have a similar question regarding particularly the FO2. I think this one's maybe even trickier than explaining how errors are measured. I'm someone who thinks a lot about FO2 and thinks a lot about reactions involving FO2. So for me, it's really critical to understand not only what the reported FO2 is, but to understand what that means. And I think your attack in general of saying, okay, we're basically trying to collect what is reported in the literature. I think I would agree that that's like a really good way to go. So you're not trying to add another level of interpretation to the data. But with FO2, I think sometimes it might be necessary to do that in some cases where it's just reported as a value. So this could be as simple as saying whether it was buffered by a solid buffer, what that was. If it was not buffered, just what this is based on. Some people might say, oh, we think our piston cylinder is about NNO, and so we think these are all around NNO. Or if they calculated it, and then how. So is it based on iron and F versus FeO activities, SiO2 activities? All that information would be really great, because then when I'm collecting a ton of this data together, I can go through and normalize them into one method when possible. That way I can compare the data more easily.
Yes.
Again, Kyla, that goes to the fields in a way that I think I will answer that with the answer to the same answer to the fields comment, that what we will try to do is we will give I don't know, star or something next to the analysis, so you can very briefly tell your way of what you are reporting. Is it dependent on the capsometer? Is it dependent on the physical buffer you use? Or is it dependent on your experiments you have been running the last 20 years? You know that your piston cylinder at that temperature does that kind of a thing. So that, we will give people a chance to very briefly tell what they are doing so that you will know without digging again into the paper. Does that answer your question?
Sure. Yeah, I think it's a good idea. I might suggest that could even be added as a recommended metadata, just to flag that it is a really important thing in case someone was entering their data and not realizing that they really need to add that info, that if they have it, please add it, kind of a thing.
Absolutely.
You hit on it, all of the 10 or so listed metadata components. You hit on the one that is probably achieves mediocrity at the most consistent rate.
Yeah, it's probably the trickiest. Yeah.
So it's an area where I think just reporting buffers is probably not sufficient in the long term. We need to get some real numbers in there. But that's again, this won't happen. None of this sort of thing is going to happen if the review community doesn't recognize the need. So that's part of what we need to talk about here. I see a question. Xiaofei, is that how you pronounce your name?
Yeah, you're good. Hi, thanks for putting this together. I just have a thought. I like the idea you're requiring people to put in metadata. I also wonder if you guys have any plans to require, set a bar to say, just when I was looking at GeoRock, way back, just trying to compile data, you get whole rock composition that adds up to be 30%. Or sometimes you look at a published paper that uses this database, you look into it, they would use this 30% total. And some of those composition totally doesn't make sense. And I feel like from the organizing standpoint, it could help the community if maybe you have some rule to say you need to pass these standards in order to, or at least flag some data that say, caution, this doesn't look right. I think on one hand, we don't want to get rid of any data that's there, but on the other hand, I know people out there, it's a world of data science and people look at the number. Seems like we could use more caution. I don't know if you have any thoughts about encouraging that.
Well,
Go ahead, Roger.
let me cut in just for 10 seconds. I've been doing this for 40 years. Of course, I have an opinion. So I'll let Gretchen say her piece, and then I'll fill you in on what my hopes and desires are for that issue.
So one of the things that, as in the case of, for example, Erica was entering the data before studying that data, we go to each paper and look at their analysis, and sometimes even the journal might have passed that, limit. Say that, okay, 90% total for a CPX is good, but we don't always, if we see something wrong and if we think that the people won't be happy with that data, even though journal passed it, we won't be putting it there. So it goes through a little bit, even though it is a reference, we are publishing the material that go through a peer review process. Within that peer review process, we saw so many times that people, spell out things wrong, or there are 2 zeros added accidentally. So we needed to go through, we don't just suck the data from there and dump it the way it is. It goes through a little bit our filter too.
Well, the other thing that we do is if we see something like an experiment that was supposedly done at 2000 gigapascals, we send an email to the author and hope they're still alive. And then when sometimes, but Phil will appreciate this. I was giving a poster about this stuff at AGU, and a student came up to me and said, "Oh, Roger Nielsen, I didn't realize you were still alive." So there were worse answers to get. But speaking of that, the new version of the template actually has a column for putting that sort of information in, basically flagging an experiment where there's inconsistencies in there are 2 different values reported for the same thing in a paper, or where they're the 2000 gigapascal problem, where clearly something is wrong. And hopefully you can get back to the author or somebody who would actually know how to fix it. But Paula's just put together an updated template. If you look on the website here, you can download. Right now, if you click on this, you'll get the template that was put together in March. Paula just sent me one this morning. This morning, that has some new stuff in it. Below that is the contact information. If after this presentation, there are questions you have later, you can just email Gotcha or I, and we'll follow up on that. Should I go ahead and go through because this sort of transitions into the template. This is the fun stuff. Let's see. What's happening, Lucy?
Sorry?
I'm trying to show the template. Let me try once more.
Using the download on the webpage, you mean?
No, I've got the template. There we go. So this is the new template that we've been working on. And if you take a look at this, there are a couple of things that are straightforward and a few that have greater level of detail. There's a README tab, which I'll let you look at later. There's a list of laboratories. Notice there's an ID number attached to the specific institution. If your institution is not on this list, you can actually just type the name in and we'll deal with it later. The reference is where the data was published so that we can access information that might not be in the template. Okay, the most important parts are from experiment on over. Two things to note. One is if you hover over the little red tabs, this will tell you what actually is in each of these columns. So this is the experiment tab, which is the experimental conditions. The first is an index number, which can be any number, but has to be the same for every component of the experiment. So that if you've got experiment H1, that's the name, H1, the author ID, which is described here as in the publication. The index is just a number so that when you go to different tabs, it will link those analyses. So that could be any number you want for the template, 4,000. But the point is that that index number needs to be the same for both the run conditions as well as when you go to the bulk composition, the liquid composition, and then whatever other phases. So let's go back and go through the experiment part. So this is the index which ties all the experiments together, the name of the specific experiment, an author ID, a lab ID, and then this ties into what we were just talking about, is that this is notes that will go into the database. It's a place where you can flag where there might be problems with the paper. You could put in here that you corrected the 2000 gigapascal number and how that was done. So you can actually flag corrections that you're making based upon contact with somebody who actually knows what's going on, or based upon a correction that was published in the experiments. So this was added specifically to deal with some of the questions that were just raised. The rest of these are fairly straightforward. The important thing to note is that under most of these columns, there's a specific vocabulary. Was there a question? Okay. So if you hover over the corner, you can see what the possibilities are. There's also tabs along here which contain more information on the specific vocabulary. And again, if you're doing something innovative and the capsule material is not listed here, then you just have to let us know. And we will obviously this is going to have to, it has evolved with time. So going back to the experiment, container, whether it's doped or undoped, pressure, the error in pressure, FO2, and this goes back, if I ruled the experimental world, I would make sure that these are all actually just numbers, not a buffer. Temperature 1, and this is somewhat tricky. We added this because of the sort of bimodal distribution of experimental approaches. T1 is actually references the superliquidus temperature if you used one. So this describes the case where you make up a starting composition, you raise it to a temperature to convert everything to glass, and then drop the temperature to a run temperature. And that's how fast you do that is the trajectory. T2 is the actual run temperature, and as well as the time at T2 and the error in temperature. Okay.
So in a way, it is designed as for dynamic versus directly equilibrium crystallization experiments, that where you go to the temperature or you go up and then drop and wait there. We have some recommendations in chat, and Dante needed to leave, but he left a recommendation which probably we can cover at the end of this.
Okay, I'm not seeing chat for some reason, but that's, hold on. Okay, are there questions about this? Can you tell me, Gretchen, if there's any questions in the chat, in additional?
No. No, the questions are how the data should be ranked and rated based on the sample size, based on how many crystals you analyze. But we can talk about that once we, after upload the data, we can extract that in the Excel form and we can do some sorting. We can explain that at that time.
Okay, we have a question from, is it Dong Zhao?
Yes, that's good enough. Thanks. Can you hear me?
Yes.
Yeah. So I'm not an expert or petrologist. I do a lot of mineralogy and crystallography work. And in our field, basically, we have something similar to this one. We call it CIF, crystallography information format, something like that. So it's also a very long table that contains all the information that's related with crystallography stuff. So the good thing with that data format is that we have some software that checks its self-consistency. For example, if you put your data and the chemical formula doesn't match the density of the material, it will give you the warnings of that. So the software will give warnings like that. So I was just wondering, are you like some people asked earlier, so are you going to also have some software to pre-check the self-consistency of the data sets, or it will be revealed by human being instead of some computer code?
It will be both. The challenge with the trace element database is that you can't, it's not set by mineral formula. So how do you know if your trace element partition coefficient is wrong? It doesn't, the trace elements don't add up to 100 and they don't behave according to a formula. One of the self-checks we do is there's a column for basically for like the liquid composition. Of course, it's way too small. is what you can do is you can actually sum all of the oxides and see how whether they are similar to what's in the publication.
I think the question is a little bit from a mineralogist versus petrologist standpoint. When many mineralogists, when they publish their tables, they don't always only give the aluminum O3 in the 8%, but the aluminum in the tetrahedral versus octahedral side kind of a thing.
Mhm.
And to be able to do that, there are programs out there for different, based on this much of an oxygen for farmland or mica and this and that. And that program itself actually can tell you that, no, this is not a good mica analysis. Don't put it in. We do not have that in the system yet, but we are hoping to incorporate. At this point, we are only reporting the oxides, not the, other than the trace elements. Obviously, we report the trace elements, but we don't report the tetrahedral or octahedral side. I don't know if that was your question.
Okay, yeah, thanks. Yeah, so I have actually another question, slightly related to what Roger comments earlier, because I'm personally, like Roger just gave an example about the data collected at 2000 GPa and etc. Yeah. So because I myself do a lot of high pressure work, we don't typically do 2000 GPa, but we typically do experiments like 100 or even multiple hundreds of gigapascals. So I was wondering, I mean, how you guys would evaluate such kind of a little bit exotic data. So for example, if I manage to measure some elemental partitioning at, like, for example, 100 GPa, will it get additional concerns, or will it be rejected by the database because it's already out of the range that people usually do, or you are still kind of welcome such kind of exotic data?
We certainly will accept anything that is high pressure. If it's phase equilibria data, that's useful for, I mean, Paula might want to kick in. If you're looking at, 200 gigapascal and you've got multiple phases that are in equilibrium with one another, that's going to be important for being able to develop models of high pressure phase equilibria. And we do have high pressure results, some of them are almost that high. I don't know, Paula, do you want to say from the perspective of Lepper about how broad the net is for the data that Mark is interested in modeling for phase equilibria?
Yes, it's completely broad, although we would tend to stick to actual natural compositions. Yeah, so we're not.
There's a question.
Binary phase diagram. Yeah, that and it wouldn't really be possible to enter those kind of data. Where as long as you've measured the major element composition of the phases involved, then the fact that it's exotic to be picked up, it can be filtered. And if people are only interested in not exotic in any sense, whether that's compositional or PT conditions, they will be able to filter the data and just get what they want. And if they want something that's more exotic, like higher pressure or whatever, they can do that as well. So.
Exotic is in the eye of the beholder.
Daniela asked a question, and Phil wanted to created more questions. So Daniela was asking that, do you think it would make sense to include a rock type category as well to this? And Paula, you answered that yes, there will be a rock type part in the database, but we need to remember that these are the experimental database, and there Phil asked that is it natural or synthetic?
2 fields and I forgot. Yeah, so there is one that we need to actually put in the template, and we will, but up to now it hasn't been populated, and that's why it got forgotten.
Along those same lines, one of the points of discussion this sort of addresses some of what Phil's talking about. One of the things that has not been part of the experimental database has been the source of the starting composition. So that's something that we've been talking about to try to tie, for example, the LEPR or Trace D's starting compositions to, was it Hawaiian basalt that was used, or was it a meteorite? It's aligned with trying to put a rock type in, but it provides something in addition. So that hasn't been done, but we've discussed that as a future direction so that people could, sort for all of the experiments done on Hawaiian basalts and use that as a metric. But we can't do that right now.
Or more importantly, if the people are using natural samples as a starting composition and get rid of the seed material versus they make up their own material. Our goal was to be able to, often in the tables, when you list your starting composition, if you are using, for example, somebody else's basalt, you give a reference to that. Our goal was to be able to go back and add that to the database so that we can give a reference to the initial, where that initial basalt is taken. I don't know if that's answering your question, Phil, or confusing you more.
No, that answers the question. Thanks.
Okay.
So let's go on with a couple of the more technical things that people have asked before and are becoming increasingly important, and are important for both the Trace Ds and particularly for LEPR. So let's go down through. This is everyone can still see the template in the liquid tab. So what we have is, for example, the analytical ID. It refers to the analysis ID. So that if you want to report, you did 4 analyses and you want to report each one of them, then you can report them 1 through 4, and then the total number of analyses would be 4. So this provides you the opportunity, ultimately going in the direction of having this aspirationally tied to a backscatter image where each of those analyses was pinpointed as to where it was from, and you can look at where the liquid was. If you want to do things that are all more than 100 microns from a crystal face, then you can do that. We can't do that quite yet, but that's sort of aspirational. And the rest of these, for each element then, there's 5 entries. One is the concentration, the error, the method of analysis, what units, and then is basic. The SiO2 standard, for example, in this case, it's just the ID of the location where the analysis took place, what the lab is. And that can be pulled out of the lab IDs, but if your lab is not there and you're just filling the template in to get it up, you can just type the name of the lab in and we will figure it out. There's not a reason to delay doing anything. In terms of things like titanium, most people, a lot of people, analyze titanium both by microprobe and ICP or SIMS. You can have multiple entries for specific elements. It doesn't stop you from doing that. Do you want to comment on that, Paula? Because you had made a note about that in the email you sent me this morning.
Yeah, so if you analyze the same species by more than one method, just duplicate this set of 5 rows. There's another template where, another tab in the template. If you want to do it by columns, you can. But anyway, you would just duplicate this and just put the second method. Just make sure you put them in order so that you can sort rows if you want, but you need to have them in the same order. So you could have all of the values and then all of the methods, or all of the errors and then all of the methods. But obviously it's going to be a little bit easier if you keep the method that corresponds to a particular value closer.
Yeah, there's no magic to the, you just need to make sure that if you have an entry for TiO2, that you have to have information for the other 4 components of that.
One thing, Roger, before you pass to the pre-compiled spreadsheet, there's a question from Dante before he left. He said he was asking that could or should be ranked the data in terms of 1, 2, 3 based on the sample size, number of the analysis of the solids and liquids. So low end should be the lower rank kind of a thing. But I agree what Prue was telling that there are experiments that we have only 3-4 crystals, especially in synthetic systems. We analyze the crystals and we analyze everywhere in the melt, and there's perfect equilibrium even with 3 crystals. So I think our goal as a template is not necessarily make that judgment. It is up to people, as long as, because when they are publishing their data, they need to pass that level of explanation to the journal anyway. So I don't think that it is our job. It shouldn't be. It should be, our job should be transparently give you everything, how many number of analysis for each crystal is, and then from there you can take it or not like it.
Yeah, so what we're trying to do is to reproduce what the authors wanted to be published, so long as they're not trying to hide something, which sometimes is apparent when you're reading the article, is that if they don't want it to be publicized that the analyses were done at 3 different labs, the obfuscation is not unheard of. But I have a question for Phil as to how he would answer this, that I've gotten questions. So what is 0? It's not detected is not 0, right? Is below detection limit 0? Or is only 0, 0?
Are you trying to catch me up here? There's actually an interesting article by someone at the USGS who had a problem with this exact topic, and has written a book and an article, and thinks that any of the techniques where you incorporate values below detection are cheating and maybe fraudulent. I would be happy to send that to you, and you can pass this article to whoever you like.
I would love to see that. The reason I brought this up is that in a previous gathering of experimentalists, and we were 4 years ago, I think it was, I asked this question to the group, and Mark Hirschman and Tom Sisson practically went over the table after each other as to what each one of them thought about the answer. So I posed that to you as a potentially divisive setup for you, Phil. But if you send me that article, I'd like to see it, because that's a critical question of how we deal with the data set with trace elements, is how do you report that? And often in the papers, it's not clear even what the author wanted.
Yeah.
I'm dealing with, this is Steve Keen. I'm actually here because I'm working with a different community that's doing something similar with EarthChem. And yeah, this has been an issue as well. I found a number of data sets where people basically, if it's below the computed detection limit, they just throw it all away. So you end up with gaps in the tables. And so there's just nothing reported at all. And as a community, we've kind of settled on, no, this is a problem, right? And so the philosophy has been just always report all of the measurements. And then, yeah, if there's, detection, you're getting close to detection limits and whatnot, well, we need to know what the detection limits are too, right?
Right.
So you have to report that as well. And then the end user can decide what's fit for their own purpose. And if the data has been censored in advance, then there's no chance to ever do that.
Right, but that, as a transcriber, it puts us in a difficult position because you can't always tell unless you understand. But it sounds like an excellent topic for a AGU special session on, nothing bothers me. But this is something if it's below detection limit and that all they do is say BDL, we can't put a number in there. We have to leave that blank because there's no other alternative. You can't put an alphanumeric in to deal with that. So these are all issues that come up when we're doing this that I don't have an answer to. But if Phil has an answer, he's going to send me. Cool. The rest of these should be fairly strict. There's nothing magic about these elements. If you don't have data for beryllium, you don't have to have these don't have to be there. So there's nothing magic. You don't need to be dealing with this huge spreadsheet of empty stuff. And as Paula said, here's just a blank sheet. What you need to do, if you've got clinopyroxene and liquid, then you just rename this clinopyroxene and you use the phases vocabulary to determine what's allowable and what's not. And this is the full name, and this is the acceptable abbreviation that you use in the phases present for the experiment section. Right there. Okay? And there's nothing magic about how it's put in. This is an example where you can put the phases in horizontally. And Paula, basically, if you put phase here, you can put all of the data in the same tab. So there's nothing magic about the organization. And the most important, as Erica would tell you, the most important thing is for the author of the data to be the one doing at least the first draft of the template, because they know the data, they know what they want to present. Once that's in, it's a relatively easy job. If there's small tweaks or errors or somebody puts a comma instead of a period, we can fix that during upload. And again, here are all the device vocabulary, the capsuled vocabulary, the phases, techniques, units, etc. But again, the important point is that the person who created the data is the one who is best able to fill the thing out. For those of you that have graduate students who are doing this sort of thing, it's useful to give them this template as they're beginning their experiments so that they will have an idea of what things they need to put in to the template. They can use this. One of the things that Mark Yorso is doing right now is he's trying to develop a Jupyter notebook that will assess the data. I'm not sure, I don't think Mark's figured out what he's going to do with that yet. Has he, Paula?
Yeah, long term, we know what we're going to do with it.
Okay, but that's something that will be future development. The most important thing right now is to get the information up and into the system. Are there any questions about the template? This is the sort of thing you sort of need to download and take a look at, and then you can follow up with questions to us. Read the chat. Yeah, I can't find the chat for some reason. Gotcha, can you unmute?
Roger, we should pass the pre-compiled spreadsheets. We have only 5 minutes left.
Okay. So here, this is what a a typical template would look like when it's filled in. This is from Alex Iverson's data. It was published this last year. Again, there's nothing magical about this. Notice that it has an index that ties together. Notice that the index and name here are the same as they are here for Amphibole, etc.
And these sheets are at this point 2 years old, and we are
Oh.
already replacing them.
But actually,
So that
that's, I need to move down to here, there. So this, for example, these are the full mineral spreadsheet. So if you have a student who is working on plagioclase, you can download all the plagioclase data in TraceDs that as of 2 years ago, using this DOI. So if you're teaching a class on trace element partitioning, these are slightly older. We're going to be uploading new ones for Amphibole, clinopyroxene, and garnet in the next month or so. That's what Erica has been working on. So these are going to be uploaded or modified in the next month. But these, for example, if you look at the clinopyroxene data in the database, there's over 900 experiments now on clinopyroxene. And based on the results from the last couple of years, the data for most major minerals is increasing about 5% per year. So that is a huge job. It's the sort of thing where no individual can transcribe that much data in any given time. That's why we need the community really to work on this.
Roger, could you please go to the EarthChem Petrology site so that you can show where these Excels live for people to be able to download?
Well, that's what they can do that here.
They're only seeing your Excel sheet
Oh,
right
sorry.
now. They don't see anything.
Hold on.
I can share my screen as well if that's easier.
Yeah, why
Yeah,
don't you-
probably it's better, Lucy.
I've got too many tabs or too many things open.
That's better. So if you are on earthchem.org and you go to communities, either from here or that button, if you navigate to experimental petrology, you can see that you can get the mineral partition coefficients and download them from here.
So these can be used for your research purposes, but as well as if you want to teach you in a graduate class, for example, using the data. They're all compiled right now, but they are 2 years old, and we will be replacing them soon.
To get back to how to tell whether there are issues or not, one of the reasons why we did this was to examine the context of the partition coefficients. With the major elements, you can see if they add up to 100, that you can also do the mineral structural formulas. With the trace elements, the only way to easily or readily find errors is to look at the trace elements in context, to plot all of the zirconium data together and see where there are outliers. And then you can go back and check to see if the outliers are outliers because the data was different and they stay, or whether there was a typo during transcription, or equally likely a typo at the time that the data was published. So these, at least if you're working on any of these 7 major minerals, these are a good starting place. In the next, so Paula, you're going to fix the link, right, for the download interface in the next few weeks?
Yeah, in the next 2 weeks.
So Lucy, if you could scroll up just a little bit. There. Yeah. So eventually that search the library, that leper tab, Lucy and Paula will be able, should be able to go through that and search the database. So that, is that going to go straight to the existing TraceD search engine we've got going, Paula?
It's going to go to a copy of it that will be hosted at EarthChem.
Okay. So several of these things are in the final boarding stages, but aren't completely operational yet. It's easy for me to say final boarding stages, Paula. Okay, it's 2 o'clock. Are there, I see there's, okay. Stephen sent a reference. So are there any other questions? Did we hit all the high points? Are you completely burned out, Phil?
Just getting started.
Okay, so this is being recorded. So we're going to post this as soon as we can get the initial 15 minutes of me talking cut out. So Lucy, can you show people where it's going to be?
I was just showing that we also recommend Whitney and Evans related to a question in the chat.
Ah, okay.
So, oh, no problem. We have some recommended vocabularies on our side in terms of what to use for general EarthChem library submissions. So if you want to go look at vocabularies there, that might be a place to start. And we always would like some feedback on that. So if you have vocabulary terms that you'd like to suggest, somebody will follow up with you. But back to the community.
So once the template is done, then you can upload it. So there's the old template.
So this is,
Yeah,
I actually updated it with what Paul gave me this morning.
Okay, good.
So once you have completed your template, you can go here to submit data to ECL. You will be redirected to log in either with your ORCID if you have one or a GeoPass ID. We would prefer your ORCID. And once you are logged in, you will be redirected to an online submission where there are some standard metadata fields related to your dataset title, authors, type of data you have in. For you, probably Petrology. And just any information you'd like to give us about the related publication. If it's not already published, you can just tell us if it's submitted or in review, etc. And once all of this is ready, you can just choose your template file and either save it if you're not done or submit it. This is going to send it to one of our curators, and in your case, also to Roger to help us with making sure it's correctly put in so that it can go into Leper in a more straightforward fashion.
We can help. So yes, this is all on Lucy's side, but if you upload a template, it will go to Lucy, Gotcha, and I, and we'll take a look and check it against the publication. So we need the publication as well so that we have all of the information and cross-check the tables before it gets uploaded. Then we need to know whether it's been published, etc. But the point is, once you send it to us, then we're in communication. We'll just figure it out as we go along. There's no linear mechanism for getting this to work. Okay, there's. Leper and trace Ds, the template is very similar. Do you want to comment on that, Paula, as to how to annotate those two? There we go.
It's the same template for both, but for Leper, you can just delete all anything to do with the trace elements that you don't need. You can just, so yeah, the Leper template is just a sort of subset of the trace Ds template. We haven't so far, but at some point we can probably post, a reduced version of the template for people that are dealing with phase equilibrium and not worried about trace elements at all. So
I don't understand that perspective myself, but I've been trying to convince Mark that he can use the trace element experiments for his phase equilibrium calculations. But the conversation is, at what point if you're using doped compositions, then there's a threshold for how much doping actually changes the phase equilibrium, but that's a separate conversation we've been having. But theoretically, of course, if you're analyzing natural levels of the trace elements, there's no reason you can't use it for phase equilibrium. An interesting thing Mark discovered was that the typical liquid compositions represented in the trace element database are aligned more closely to natural lava compositions than the liquids that were originally used in Leper. So the trace element experiments actually are more natural than the Leper database. And part of the reason for that is that a lot of the work, I blame the geophysical lab myself, but a lot of the original work was done on end member compositions, and there's more of a tradition of doing it that way for major elements. And the trace elements, most of us, especially recently, have been using more natural starting compositions. So the liquids align more closely to the lavas we actually see. So I'm not sure where that's going to go, but that was something actually Mark found. And I think he actually reported in a paper a couple of years, maybe it was just an AGU presentation. Any other questions? You want to add anything, Gotcha or Erica?
I think we are out of time, but if you guys have any question, you can reach us at any time.
Even if
We just need to be able to do these workshops as efficient as possible and as much as possible, so that it's not something easy to learn in one.
You are all guinea pigs.
But thank you all for coming.
Thank you.
Thank you very much.
Okay, so have a great day, and we save everything from the chat, and so you'll be able to see your questions, comments, and everything.
Okay, take it easy, everybody.
Thanks.
Bye-bye.